So Philip is a medical doctor, trained at Kiel University in Germany and also at Tufts University in the United States. And ever since 2001, has, as a postdoctoral fellow at the time, has been doing research at Kiel University, where he's now the director of the Institute of Clinical Molecular Biology and also professor of molecular medicine. Philip's research targets inflammatory bowel disease and the functional effects by genetic signals, focusing on cellular pathways and networks of the node-like receptors and autophagy and ER stress. Philip's work regards, and if this is not right, let me know, but I think Philip's work regards the host and its gut microbiome as a functional unit to better understand the mechanisms driving IBD. So today we'll learn about how dietary factors license host microbe interactions at the intestinal barrier.
So thank you very much. I hope you can hear me. Give me some thumbs up. Is that okay? Can you hear me?
Yes.
Good. Okay, thank you. So yeah, today I will try to cover a quite complex topic, and that is the idea how dietary factors interact with microbes to have certain physiological effects on the intestinal barrier, but also on immune responses or other host physiology. And I think before we start, let's have a very general look at the gut microbiome as an ecosystem and the question, and I think that's something that really came up 10 years ago, and that was shaped by a vicious discussion around the so-called enterotypes, whether there are so-called preferred states of this very chaotic and very individual constitution of the microbiome in the gut. And of course, the idea is if the gut is a bioreactor where microbes also use nutritional factors as fermenters or as kind of an ecosystem together with the dietary factors, then of course, what you put in should have some kind of effect on potential states. And it's very clear that from microbes that there are some microbes that are able to consume more carbohydrate structures and the other ones more proteinaceous structures and so on. And that was actually also kind of at the first paper, the seminal paper by Per Borg and Jeroen Ras and others here in Nature in 2011, kind of put forward that there might be optimal states or equilibria. And there was a strong discussion whether this is really stable, whether these are rather enterogradients, whether you can shift between the constitutions. So the concept was completely misunderstood in the beginning and was compared to blood groups, which of course is not genetically determined. So never you will have a genetic strong constitution of the microbiome as such. I mean, you have some influence of the genetics here. But in the same year and much less debated, there was another paper which was probably as seminal that showed something very similar, where there were only two enterotypes. It was also called enterotype. And these enterotypes were clearly linked to the dietary intake. So in other words, the enterotype 2 was linked to the intake in carbohydrates. I hope you can see my pointer. Can you see my pointer? Yes? Yeah, okay, yes. Clearly linked to the intake in carbohydrates here, whereas the enterotype 1 was more linked to the intake of protein material. And I think this together was the first evidence, or one of the first evidence, like on the microbial genomics space, that there is such kind of binary or tertiary or whatever distribution here. And of course, the question now is, suddenly, it becomes a very, as a naive physician, you suddenly have, how can the host be impacted by nutrition, right? Of course, you learn that microbes help you digest things, but ultimately, there will be many bioactive metabolites coming out of such a dynamic system here, where of course the diet still influences directly or still has direct influence on the host, but the microbial network and its constitution will also have a big influence how the host is reacting under certain circumstances. I will give you two examples, one of them focusing on short-chain fatty acids, quite recent work from the lab, and the second one focusing on a long-term investment in amino acid homeostasis. Now let's turn to the first example here, and that is how microbes and microbial products actually regulate a hexokinase, namely hexokinase 2, by short-chain fatty acids or fiber fermentation, and how this actually impacts on host immunity. You all know that their hexokinases are the first rate-limiting step of glycolysis, so they actually catalyze the glucose 6-phosphate addition here. There are 4 hexokinases in humans, and hexokinase 2 is a very specific one that is kind of has limited expression. Hexokinase 1 is the most abundant one, and hexokinase 2 is thought to be stoichiometrically negligible in most conditions for this reaction, but it's increasingly recognized that this hexokinase also has other functions, such as pattern recognition, meaning recognizing microbial products with a similar chemical structure. Now, these other products kind of brought our attention to this molecule here, and that is also now, and somehow this is gone here. So this is from a recent EMBO reports paper from 2020. It is clear that hexokinase 2 has a role in the regulation of cell viability, and recognition of microbial products by hexokinase 2 can actually lead to induction of cell death by mitochondrial permeability regulation here, and this is specifically recognized in epithelial cells and tumors. I'm now showing work by Felix Sommer, Finn Hinrichsen, Jakob Hamm, and many others, and Felix now also has his own group here, and that's a recent paper that came out here where he could actually show by laser capture microdissection and comparing conventionally raised CR versus germ-free mice here in panel A, that there is microbial induction of hexokinase 2 in the tip of the epithelium in the colon here, as well as in the ileum, and that this is rather repressed under germ-free conditions, but also kind of in the crypt. So there is kind of spatial regulation by microbes in the normal gut by the microbial network here. And this is also true when you just start here from germ-free mice and conventionalize them at a later time in adulthood, and you see that in the ileum and colon tip here, there is increased expression of hexokinase 2. And Felix then also went on and tried to, in monocolonized animals here now, to bring this effect here. So you have germ-free, conventionally raised, and then trying to understand which bacteria can actually ramp up the expression here. ASF is altered Schedler flora, so that's kind of the normal flora from mouse house, and OMM is the microbial consortium here that contains certain strains here that I'm going to refer to in a second. But it's clear that fermenters such as B-theta can also induce this hexokinase 2 expression. And then as there was strong induction by OMM, and these strains are known, Felix then and the group went on to actually show that if you dissect certain microbes in this oligo consortium here, there is both repression as well as induction in the individual supernatants here from these individual bacteria. And that is kind of interesting if you can, by bacterial metabolites, actually switch on and switch off such a molecule here also therapeutically. And to cut a long story short, it was then shown that basically the KB1 is a strong acetate fermenter, whereas I46 and YL31 actually are strong butyrate producers. And if you then just take these fermentation products such as butyrate and acetate, we could then show that actually butyrate is able to repress HK2 expression, as you can see here, and acetate, vice versa, is able to induce HK2 expression. And this is also for an interacting molecule of this VDAC for this mitochondrial permeability. You also see that here that butyrate is also repressing the normal expression here in intestinal epithelial cells. And Felix then went on, and others went on here to show that there are certain HDACs, so butyrate has a known effect on gene expression, as you know, by modulating chromatin states, and there's a certain group of HDACs that is actually responsible for that effect. Now, what does that mean, and why am I showing that? Well, how is that modulating actually host immunity here? Now, we are dealing with intestinal inflammation, and butyrate is a well-known substance, and also butyrate as a fermentation product is a well-known protective substance and has also been used in clinical trials, although the I would say, the smell, the odor of butyrate in the end, and also the irritant effect of butyrate at high concentration, really did not lead to approval, and specifically not social approval of that substance for treating inflammatory bowel disease. But it's very clear if you give that to animals and you induce inflammation, that you have a protective effect. So what you see here is one of the classical models that we have, that is induction. This chemical irritant is called dextran sodium sulfate. So this basically eradicates the epithelium and have a strong inflammatory response. And if you then give butyrate under such conditions here, and body weight is then kind of reminiscent of the inflammatory response, meaning the faster you lose body weight, the more inflammation there is. And if you then give butyrate specifically at later points when there is a regenerative response, you can see the effect, the protective effect here of butyrate here in this model. And you can also see that there is suppression of cell death here, and that's the epithelial cells by butyrate. Now, under these conditions, we think that then the butyrate would downregulate the hexokinase and thereby dampen the inflammation and cell death, right? That would be the hypothesis. And indeed, when we then used, Felix then used the HK2 in a specific knockout model where we deleted them in the HK2 in intestinal epithelial cells, where this regenerative response takes place, the regulation takes place, suddenly the protective effect of butyrate was gone, meaning that there already is downregulation, then butyrate doesn't have any effect anymore, rendering the HK2 the culprit. So it's kind of necessary, the downregulation is necessary for that protective effect. And also cell death and other things are basically then eliminated in their significance. Now this is a big step forward, because we would now think that HK2 inhibitors, kind of inspired by this metabolic interaction here, that HK2 inhibitors could be brought forward as more acceptable therapeutic principle here to license this inflammatory response. So this is kind of how short-chain fatty acids, normally induced by fiber fermentation by specific bugs in the gut, actually can have an impact on the inflammation. So the second example is around amino acid homeostasis, and that is really something that we work on for quite a while here. And specifically, we work around mechanisms on protein energy malnutrition, how this is affected by microbes in the gut. Now, you all know that malnutrition obviously is, when you think of recent human evolution, like the last 150,000 years or so, that probably malnutrition is rather the normal state, right? So if you look back 50,000 years, probably protein availability and enough food is rather something that you see sometimes, and that the kind of surplus that we have currently, that we can just go to a supermarket, obviously is something that's just very, very recent. Now, of course, this is different in other parts of the world. When you look not only in Europe, and again, this malnutritive state is often driven by protein availability. So protein energy is the good stuff, basically, in the nutrition here. And still, it is the cause of millions of deaths in underdeveloped countries and crisis regions. But it's not only there. You specifically now also with the COVID pandemic, if you go, for example, to the US and the social problem in the US, that 40 million US American citizens are officially considered food insecure, among them 12 million children. And of course, this is aggravated by the pandemic situation, as I said. And it's not only the food insecurity, but the cheapest food that is available in such Western industrialized countries obviously contains a lot of carbohydrate and fat, so that protein energy malnutrition and protein availability again becomes a problem here. And not only there, but also when you think about disease, for example, IBD or also cancer and aging, malnutrition and malabsorption becomes an individual problem. And again, protein availability is part of cachexia, for example, in cancer, and that's a well-known thing. Now, there is many things. There is acute effects and memory effects of malnutrition. And specifically, we are specifically interested in the immune defects in this state here. And just to reiterate here, so I don't know why my whatever. So the acute effects are actually the metabolic perturbation, of course, of dietary protein going down. And you all know the kwashiorkor and other diseases linked to this very highly acute malnutrition. But they don't really die of the kwashiorkor, but they die, the individuals die because of the susceptibility to infection and the dampening of adequate immune responses. And part of that, and that is work by Jeff Gordon and others, I think has clearly been shown, is transmissible by microbiota. And of course, the antibiotic use in acutely undernourished children, for example, is the only clinical thing you can do to actually rescue them, apart from, of course, getting them proper food. Again, I've alluded to Jeff Gordon. I mean, many studies here, and that's just one of them, using FMT, fecal microbial transfer, from discordant twins, for example, one of them having kwashiorkor, the other one doesn't, clearly showing that there is certain bacteria that are higher abundant in this state, and this actually leads to transfer of the phenotype when you bring that into gnotobiotic animals. And this really has led also to insights that kind of change the way how clinical management is done, also in underdeveloped regions now. And this was a recent paper published in New England Journal paper, also under the guidance of Jeff Gordon here, where this knowledge was actually transferred into specific dietary interventions, where there was just the ready-to-use supplementary food was kind of compared to microbiota-directed complementary food with very simple ingredients, just aiming to reestablish the balance here and to overcome these outgrowth of the pathobionts. And this is probably also why this is considered a landmark study here, really has shown that by thinking about what is needed to outgrow certain bacteria and bringing that in the rational way into the children in that case actually alters the way how bacteria look like, and also alters the way how weight is gained and other phenotypes are overcome. Now, what we are interested in here is how do certain specific metabolites in the condition of specific malnutrition actually impact host immunity? And that is really coming from also a kind of chance observation that we made together with Josef Penninger some years ago, where we saw, and you all know ACE2 now for other reasons, because it's also the receptor for SARS-CoV-2. But at that time, what we made as an observation is that ACE2 knockout mice actually have very strong barrier defect, and they have an overgrowth of certain bacteria that you see here in red that are can overcome a tryptophan deficiency here. And really the effect is that there is a local missing effect of the absorption of certain amino acids in intestinal epithelial cells. There is an energy deficiency in intestinal epithelial cells, and that barrier breach and the downregulation of certain antimicrobial peptides basically leads to the outgrowth of pathobionts, very similar to the children in Jeff Gordon's stories. And this actually leads to strong intestinal inflammation. And this inflammation is also transmissible and can be overcome if you bring back nicotinamide, NAM, as one of these metabolites downstream of tryptophan. Tryptophan is essential amino acid into these epithelial cells. This restores the antimicrobial peptide expression, restores the S6 kinase pathway, mTOR pathway in these epithelial cells. And this then brings back also the microbial homeostasis here. Now we went on, and this is with Tiro Kaneganti here, and also showed that there is a certain microbial other signals kind of adding to this pathway here. So this is not only the antimicrobial peptides, but it's really also the mTOR pathway. And this is kind of licensed by certain microbial driver and restraining signals here via NLRC3 and PI3 kinase that can basically then limit this cell survival effect. And this kind of very basic effect that also drives cell regeneration under or kind of limits or drives cell regeneration under intestinal inflammation conditions is also exploited by intestinal tumors, and to overcome basically the growth restraining signal to then have a CRC, so colorectal cancer development. So that's a very basic principle where you have this, in this case, was active energy signal leads to restraining signal, and this is controlled by the microbial sensor NLRC3. And if this is lost, basically you have aggravation of tumor outgrowth. Okay, this is the intestinal epithelium and the barrier. And we made another observation. So basically there is strong local inflammation, but we made another by chance observation here that adaptive immunity actually reacts very different. And that's, again, a story we've done together with Michael Platten and Jana Sonner at the German Cancer Research Center. And the question was, can we basically use tryptophan and tryptophan metabolites to shape immunity in the gut, but also in other places? And Michael is a neurologist, also clinician, and he made a also funny observation that so he works with multiple sclerosis, and there is a famous multiple sclerosis model. It's called EAE, experimental allergic encephalitis, which is recapitulating part of human multiple sclerosis. Basically, you immunize mice with the kind of myelin, parts of the myelin sheath, and then these mice will have an immune reaction. So there's specific autoimmunity induced by self-peptides from the myelin oligodendrocyte glycoprotein, MOG, and you need an adjuvant that breaks down the blood-brain barrier, and then you see this. So this is how such a reaction looks like, and you see here that there is a strong demyelination here. Oops, where's my cursor? You see that there is a strong demyelination under these conditions, which is driven by this kind of vaccination with self. Yeah, so it's kind of very similar to the multiple sclerosis model. And the idea was, can we somehow restrict the diet and then see whether there is aggravation or kind of enhancement or inhibition of this immune response. So in other words, are there places also, or are there dietary interventions that we can use to bring down this event? And there was some evidence that tryptophan and protein and energy availability, again, could play a role here. So this restricted diet was compared to control diet. And the real funny thing was, or the striking observation, to be more scientific, was that if you don't have protein, there is not a single T cell invading the CNS. And that was, of course, you can say, well, that's energy restriction and the animals will die anyhow, and so on. We then went on and measured by metabolomics, we measured the amino acids. And of course, it's very clear that the kind of essential amino acids are then reduced under such conditions, and it was specifically methionine and tryptophan that had lower levels here. And we then went on and asked, because we had this ACE2 phenomenon here, we asked next the question, can we mimic the protein-free diet with the tryptophan-free diet? And that was the case. So basically you also can, so the brown cells here are the infiltrating cells here. And so these are CD3s here. And this is the demyelination that you can see. And the demyelination score kind of goes down to close to 0 when you take away the tryptophan. Now, does any essential amino acid do the job here? Because that would be when you look at the way here, okay, you reduce protein, you reduce the available of essential amino acids, and thereby probably you cannot mount an immune response because you just need normal amino acids to overcome or to mount an immune response. And that's definitely not the case. So if you take a methionine deficient diet on the same microbial conditions, same animal house, then you see a mild inhibition here of the immune response, but clearly not the reduced to 0 clinical score here, or also the histology was also not affected by the methionine. So it's not just the essential amino acid, but it must be something different. And the next experiment was again just asking, is it necessary that we have an oral delivery of the tryptophan? Because when we think about any microbes that would contribute to that, then an IP or an intravenous injection of the tryptophan would overcome the kind of deficient situation, but the intestinal microbes would not see the amino acid, right? So that exactly was the case here. So that's a bit. So on the left side, you see here that's the normal condition plus tryptophan, and then there is the inhibition. And then if you give back on a later day after the vaccination here, if you then start feeding the animals tryptophan again, then there is an again mounting of this autoimmunity, as you can see here. Now, if you inject the tryptophan intraperitoneally, so thereby kind of circumventing the microbes in the gut, the tryptophan inhibitory effect stays basically the same. So the oral tryptophan was needed for neuroinflammation, and there was some kind of idea that there is a direct mucosal licensing effect here of the tryptophan and/or metabolites. And then we went through, and there was work done by Jana Sonner here. She basically went through now every knockout model there was where certain parts of tryptophan can also be sensed. So there's, for example, the GCN2 kinase, which is the major kinase that links amino acid levels with the mTOR pathway. Long story short, no effect. And but what was then very evident is that when we did the same experiment under germ-free conditions, the suppressive effect of the tryptophan deprivation was more or less gone. So meaning microbes must be present. There was a slight effect on the clinical score. You see here there is a kind of slightly diminished clinical score here. Oops, there's a slightly diminished clinical score. But if you take the hard score here, demyelination in the CNS, then demyelination in the absence of microbes is more or less the same, regardless whether there's tryptophan present or not. Meaning that microbes are involved in producing something or eating away something that is, or is kind of involving the microbes to produce a protective factor here that suppresses the inflammatory activity. Elsewhere, you need to see it in the gut, but the inflammation is elsewhere. And I think this is a very crucial finding, that you can license systemic immune responses by having microbial product in the gut. Yeah, so that's a very, at least we thought that this is a very seminal finding. We then tried to find this factor, and we clearly failed. And this is also why, after a long fight with a journal, this was not published in the journal without the communications, but this was published in the journals with the communications, because the short letter journal wanted to have the factor, and we just tried to do many, many things, including using aryl hydrocarbon receptor knockouts, which are known to actually send certain tryptophan metabolites from the microbiota, no clinical effect. Feeding animals with certain known tryptophan metabolites from the microbes, no clinical effect. And the only thing we could actually offer is that, yes, under these conditions, the microbes in the gut look very different, completely different. And when we do metagenomics and prediction of metabolic networks, then we see certain things that are over or underrepresented under such conditions. But at some point, we found that the finding itself was so fundamental that we wanted to publish that. And so we are still left here now with metabolomics and metabolite screening to actually understand this immunosuppressive phenotype, which we think is very important, because it would mean a completely different new avenue into using diet microbiota interactions to dampen autoimmune responses in the brain. And this is something that also, where kind of protein restriction diets are actually also in clinical trials now to suppress inflammatory activity. With that, I'm at the end, and I hope I've shown you in 2 examples that many different dietary factors and their interaction with the gut microbiota may have a strong impact on many host responses, one of them being very local, like the intestinal inflammation I've shown you in the first one of them being completely distant and behind an immunoprivileged barrier such as the brain. And it also tells us that even with simple things such as tryptophan that we kind of think we understand, still new pathways can be defined, and we are still struggling with that. And the key questions really are, can we use these metabolic principles of this co-metabolism to shape either the microbes or use postbiotics like metabolites of the microbes in a very rational way to enhance therapies or to even use it as a therapy. And I think this really is not fantasy. And of course, I have to thank many people involved here, and I thank you for your attention.
Thank you so much, Philip. Great talk. Any questions, please? You have a question, come forth. I have actually one question regarding your first part of the story with the exokinase 2, that you show that the suppression is important for inflammation and colitis, right? Mm-hmm. How does this, how does the expression of HK2 work in a model like that is prone to colitis, like an IL-10 knockout model?
Yeah, that's something that we're currently looking at. Can we actually modulate that in a sense, right? And that's also, so there is upregulation of expression under inflammatory conditions. So in a way, it's something that can also be used to block that, right? And that's also the idea that it is present under inflammatory condition, and the modulation of that by certain inhibitors, not downregulating the expression, but rather inhibiting the activity or stabilizing the molecule, it could be a new way, because obviously the butyrate, smells horrible, and it's an irritant acid, and that was also the limiting factor. So there were clinical trials using a butyrate at higher concentrations with enema, so basically blowing that into the colon. The clinical signal was great. I mean, dampening inflammation was great, not only in mice, also there was some signal in humans. But the problem really is the irritation that the acid is then basically relieved after a few minutes. So the patients cannot hold this therapy long enough so that and then you have the butyrate basically in the bed or in the trousers. And that is obviously something that was a very limiting factor. Also the IP situation is a limiting factor for pharma development here. But butyrate, of course, it's just one of the principles. I'm not saying that butyrate can be now used to heal ulcerative colitis or Crohn's disease, but I think it's interesting that you can disentangle the kind of just the overarching role of butyrate as an HDAC modulator and modulator of gene expression. You can bring that down to certain molecules that might be needed for the protective effect, as we've shown, and where very specific and targeted developments now could be used to overcome certain aspects of inflammatory diseases.
Thank you. We did have one question that you already replied to, actually, and you answered that, asking if it was the acid that you used in that experiment. And there's another question from Carlos Vot. I'm going to read it. Thank you for the interesting talk about HK2. What does the induction HK2 expression by bacteria mean for the inflammatory response? Is it pro or anti-inflammatory? And do you know what the microbial ligand for HK2 is?
Yeah, we would think that this is rather a pro-inflammatory response, as I pointed out. And the microbial ligand for HK2, there is some idea, but it's not really clear what is actually sensed. It could also be just the levels of glucose that is sensed, or other sugar ligands that are kind of cell wall components could also be sensed, and that's still a bit open, I would say. So that's I would say that's ongoing discussion. Of course, I mean, kind of co-metabolism of certain sugars is also important in not only in humans, but also in many organisms. And at least the notion is put forward that these hexokinases that are not the classical hexokinase could also sense this kind of competition for certain sugar residues, or could also sense other sugar metabolites. But there, the last word, I think, is not spoken, what they could actually sense also in addition.
Okay. Once again, thank you so much.